Good morning, students. Hope you read Translation Part 1. In last lecture, we saw how the protein biosynthesis takes place and steps of translation, that is, initiation, elongation, and termination. Learning objectives for this lecture. After the lecture, you should know what are the differences in eukaryotic and prokaryotic translation, how the regulation of translation occurs, post-translational modifications, and inhibitors of protein synthesis. So, differences in translation in eukaryotes and prokaryotes. Mainly, there is a similarity between prokaryotes translation and mitochondrial translation. They are almost similar. And DNA ribosome differences, we know that it's in eukaryotes ribosome is 80S and in prokaryotes it is 70S. Initiating amino acid for eukaryotes is methionine and for prokaryotes it is formylmethionine. Major difference arises in initiation factors which are very important for the initiation step of translation. So in eukaryotes, there should be minimum 9 initiation factors and in prokaryotes, 3 initiation factors are there. Elongation factors for the elongation steps. So in eukaryotes, they have EF1 alpha, EF1 beta, EF1 gamma, and EF2, while in prokaryotes they have 2 elongation factors. So these were the major differences. And the tetracycline effect is also no effect on the eukaryote translation, but protein synthesis is inhibited in prokaryotes. Now we will see the regulation of translation. So basically regulation occurs by eukaryotic initiation factor 2 preventing 43S pre-initiation complex formation. So what happens in this initiation factor 2 is activated by phosphorylation of alpha subunits which binds to eukaryotic initiation factor 2b and inactivates it. So this prevents 43S pre-initiation complex formation and stops translation. And the other regulation is through EIF4 complex and preventing 48S initiation complex formation. So in this initiation factor 4E binding to mRNA cap to stimulate translation. Examples for example, insulin and mitogens. So how it acts or regulates translation. Now, we will see post-translational modifications. The newly synthesized proteins after translation are called nascent proteins or polypeptides, and these are inactive biologically. So these become active to perform their function only after they undergo some specific modifications. So these are called post-translational modifications which make it active. Now we see different types of post-translational modifications. We have reversible and irreversible both types. In reversible, there will be modifications of disulfide bridge, glycosylation, phosphorylation, acylation, N-acetylation, ATP ribosylation. And in irreversible, we have proteolysis, ubiquitination, lysine hydroxylation, proline hydroxylation, and methylation. So, major post-translational modifications we will see. First is the protein folding. with the help of chaperones to acquire an appropriate three-dimensional structure to become active. Chaperones help to produce correct spatial arrangement. Examples of chaperones are the heat shock proteins. This folding is largely mediated by non-covalent weak interactions, and the proteins that do not fold properly and become non-functional are degraded and can lead to prions diseases. Second is the proteolytic cleavage which occurs. So usually newly formed proteins, or we can say nascent proteins, they are larger proteins than their mature functional counterpart. They are called as pre-paraprotein, and they undergo cleavage to become smaller and active. So the post-translational modification occurs through the proteolytic cleavage. Then third is the modifications of amino acids. Modification of amino acids can occur through gamma carboxylation, hydroxylation, phosphorylation, glycosylation, and formation of disulfide bonds. So in carboxylation, what happens? Carboxylation of glutamate residues in coagulation factor like prothrombin, which is necessary for their function. In hydroxylation, proline and lysine in collagen undergo hydroxylation to form hydroxyproline and hydroxylysine, which are again important in maturation of collagen. Phosphorylation of serine, threonine, and tyrosine residues occurs by specific kinases. Glycosylation, it occurs by addition of oligosaccharides chain to specific amino acids, residues of the polypeptide, and is seen in glycoproteins. And the other modification is through the formation of disulfide bonds. So disulfide bonds are formed between different cysteine residues within the same polypeptide intrachain or in different polypeptide chains, that is interchain. Example for it is the insulin molecule. After post-translational modification, we will say inhibitors of protein synthesis. So, various antibiotics, they also act as inhibitors but selectively acts on bacterial ribosomes, that is for prokaryotes, and other translation components to inhibit protein synthesis and kill them. So, we have different types of inhibitors. There are different types for prokaryotes, eukaryotes, and there is one puromycin which is for both prokaryotes and eukaryotes. For prokaryotes, inhibitors are tetracycline, doxycycline, chloramphenicol, erythromycin, and aminoglycosides. For eukaryotes, diphtheria toxin, ricin, cycloheximide, and puromycin is for both the prokaryotes and eukaryotes. Diphtheria toxin, it catalyzes ADP, ribosylation of eukaryotic elongation factor 2 occurs and prevents translocation, and it blocks elongation. Ricin is present in castor beans. It binds to 60S subunit. It removes one adenine residue from 28SR RNA. Cycloheximide, it binds to 60S subunit. It inhibits peptidyl transferase. It blocks elongation. And puromycin, it resembles amino acyl tRNA, incorporates in growing polypeptide chain and causes premature termination of polypeptide chain. So puromycin is inhibitory for the prokaryotic and eukaryotic translation both. So it causes how premature termination of polypeptide chain occurs. So that's all about the translation. Thank you.